protein factories. So what are protein factories? The only cell organelle which is present in both prokaryotic and eukaryotic cells. So ribosomes are these small granule-like structures. A ribosome is what? Ribonucleoprotein particles. Richard Roberts, RR. Very easy to remember. RRR. That's all. Right? The trick to remember. RRR. Now, who doesn't remember RRR? The Oscar-winning song, right? What is the 70S and 80S type of ribosome? For more and more binding capacity to oxygen. Now, I can't take maths brain deep into the biology, but I can take a biology brain deep into this. Because of this ribosome, the RER, the rough endoplasmic reticulum, is able to produce protein. This structure is called as polyribosome. Ribosomes are produced and assembled where? Lysosomes, endoplasmic reticulum, Golgi apparatus, or the nucleolus. Hey guys, welcome to this class. This is Deveeka, your biology buddy, and we are going to talk about protein factories. Yes, you heard me right. Protein factories. So what are protein factories? Ribosome. Yes, it is a very unique kind of cell organelle. The only cell organelle which is present in both prokaryotic and eukaryotic cells. The only cell organelle which do not have any membrane. In the end of this session, I will also give you a challenging question which is of NEET level, and let us see whether my 9th graders are making a progress to solve NEET questions, which I'm sure you will do. Now, what are ribosomes and who did the discovery? A little bit of the history. Okay, so ribosomes are these small granule-like structures, right, which are present in the cytoplasm and mostly on the endoplasmic reticulum. Now, when they are so small structures, right, they are ribonucleoprotein particles. A ribosome is what? Ribonucleoprotein particles. Now, read out that word one more time. Ribonucleoprotein particles. Ribo, rRNA, right? Ribonucleo because the site of synthesis of the ribosomes is the nucleolus, the dense body, round dense body present inside the nucleus, nucleolus, right? So it is the site. So nucleolus is also known as ribosome synthesizing factory, and it has been produced over there and then it is distributed to the cell, right? So it has what? Ribonucleoprotein, because ribosome is made up of rRNA, two components, rRNAs and proteins, right, chemically. So that's why ribonucleoprotein. Name is justified now. Now let's see how did it start. So they are so small that it was not seen under the compound microscopes or the small ordinary kind of microscope. It required electron microscope. So when electron microscope was developed, it was first observed by George Emily Pallady, right? And the scientist who gave the term, the name of ribosome was Richard Roberts, right? In late 1950s, he termed them as ribosomes. Alright, so who all? Once again, Emily Pallady observed it via the electron microscope, and Richard Roberts, RR. Very easy to remember. RRR. That's all. Right? The trick to remember. RRR. Now, who doesn't remember RRR? The Oscar-winning song, right? So now here, RRR. Robert Richard Ribosome. Termed ribosome. Fantastic. Now let's go ahead. What is the structure of this ribosome? So the ribosome is made up of two subunits. Okay, there are two units or two subunits. It is membraneless spherical bodies, round membraneless. They do not have membrane, as I told you, like your endoplasmic reticulum, mitochondria. They have membranes, right? This is having no membrane, and that's why it is also seen in prokaryotes, right? So prokaryotic cells do not have cell organelle, but they have ribosome, because we say prokaryotes do not have any cell-bounded cell organelles, right? But it has ribosome. It is made up of two subunits. and the subunits are placed on one top of another. One is the larger subunit, okay, one would be the larger subunit, one would be the smaller subunit, and each subunit is made up of what? It is made up of ribonucleic acid, RNA, right? Like DNA, deoxyribonucleic acid, this is made up of RNA, rRNA, right? So, ribonucleic acid and proteins, which are called as ribonucleoproteins. They are made up of ribonucleoproteins. Now, I told you why it is called as ribonucleoprotein. Because of synthesis of this proteins, this rRNA happens where? In the nucleolus. That's why. Okay. So now, are they present in all the cell sites? I told you they are present in eukaryote and prokaryote. No. Right? Some of these cells, special cells like the sperm cells, they do not have ribosome. The mammalian RBCs which are matured, they do not have that, right? So RBCs, the mature RBCs do not have mitochondria, they do not have Golgi apparatus, they do not have endoplasmic reticulum. So far so, why? They do not have nucleus as well, right? So all of this is eliminated so that the RBC cells can be modified to have a special biconcave structure for more and more binding capacity to oxygen, right? So they are not present in mature sperms or your mature RBCs. Now, where are they present? They are present in the cytoplasm, right? And some of these cells, organelles, like your mitochondria and chloroplast, they have their own ribosomes, okay? They have their own type of ribosomes. Why? Because plastids, like chloroplast and mitochondria, have their own DNA. They are the semi-autonomous organelles. That's why. Alright. Then the cytoplasmic ribosomes may remain freely in the cytoplasm, or they are attached to the ER, endoplasmic reticulum, because of which the endoplasmic surface appears to be rough, and we call it as rough ER, rough endoplasmic reticulum. Because of this thing itself, because of this ribosome being embedded on the endoplasmic reticulum, it can involve itself in protein synthesis, right? So, ribosomes are attached to the membranes of endoplasmic reticulum by the larger subunit. Larger subunit is attached and smaller is outside. That's why they appear like a rough structure. Now, how do we name it? And actually, before I tell you about this, ribosomes are of two types, majorly two types. The two types of ribosomes are 70S ribosome and 80S. I'll explain you shortly what is 80-70, right? So, those 70S is prokaryotic type of ribosome, 80S is eukaryotic type of ribosome. Now you are hearing this word letter over here, S. So why it is called as S? So basically this stands for a unit called as Swedberg unit, right? What is a Swedberg unit? Swedberg unit is used to understand the sedimentation rate. Right? So the size of ribosome is measured, how it is measured by the sedimentation rate. What is sedimentation? Now imagine you have taken a centrifuge, right? Centrifuge has shafts like this, and to this we have attached the tubes. And what does this tubes have? This test tubes have the cell components. Ribosome, you have extracted isolated ribosomes, and the ribosomes are embedded over here in this solution. Okay. So now when we are doing the centrifugation, they will start settling down. That is called as sedimentation, right? And what happens usually? The larger heavy particles will settle down fast. Yes, so 80S ribosomes would settle down fast. Why? Because the Swedberg unit is actually indirectly measuring its density and size as well. Okay, it's measuring its density and size as well. So this S stands for what, bacha? It stands for Swedberg unit. Okay, it stands for the Swedberg unit. And how is it measured? By the sedimentation rate. Now, what is the 70S and 80S type of ribosome? Let's understand that. So 70S is present in prokaryotic cells. Where are they present? They are present in prokaryotes, and inside of the cell organelles like mitochondria and chloroplasts like plastids, which are the semi-autonomous organelles in eukaryotes. So 70S is basically a prokaryotic type of ribosome which is present in mitochondria and in the chloroplast. Why? Because mitochondria, chloroplast have its own DNA, circular type of DNA, which is again similar to the bacterial DNA, right? So that's why prokaryotic and mitochondrial and plastid have this 70S type of thing. Now 70S is made up of, as any other ribosome, it is also made up of 2 subunits. So the larger subunit is 50. Now this is possible only in biology. Right, this is possible only in biology, that 50 plus 30 would become 70. Sorry. The mathematician in you, right? The maths teacher would be like, ye kya ho gaya? Lekin ha, it's possible only in biology ki it should be 80 but it is 70. Okay, the weight comes to 70S. Right, so 50S is the larger subunit, 30S is the smaller subunit. So this is 30S and this one would be 50S in this case. Okay. Now about the ATS. ATS is present in cytoplasm of eukaryotes and attached to the endoplasmic reticulum in the eukaryotic cell. Absent in prokaryotes. 70S found in prokaryotes as well as eukaryotes. ATS only in eukaryotes, only in developed cells. Now here, what is the case? Here you have 40S and 60S. 60S being the larger subunit, smaller subunit being the 40S. Again, it's only possible in biology. Ki 40 and 60 is becoming 80 over here. Okay, so now what is the justification for this? That's the justification for this, okay. Now I can't take maths brain deep into the biology, but I can take a biology brain deep into this. Why 60-40 is becoming 80 and why 50-30 is becoming 70, right? So what is this 70S? Sedberg unit. Yes, Sedberg unit is for what? It is for sedimentation rate. That S is Sedberg unit, right? So now focus on this. This is 70S type. This is 70S type of ribosome. Okay, 70S type of ribosome is made up of what? It is made up of 50S larger subunit, yes, and a smaller subunit of 30S. Okay, what is the size? The size is measured in angstroms, very little, right? That's why 200 to 290 angstrom. Whereas when we compare the ATS would be a little bit bigger in size, yes? So 100 units ka difference would be there, near about 300 to 340 angstrom. We can remember like this, 200 and 300 angstrom if we talk about size. Now let's see, as I told you, the ribosome is made up of what? It is solely made up of RNA and protein. That's it. RNA and protein. Chemically, ribosome is made up of what? RNA plus proteins. Now, proteins can be of different size. है ना? Different sizes of amino acid chains. So, if we talk about the 50S subunit, it has near about 34 proteins. Kitna proteins? 34 proteins. And what is the type of RNA is present over here? 2 types of RNA. What types of RNA? 5S, again S being the sedimentation rate, right? And 23S. 23S and 5S is making up your larger subunit. If we talk about the smaller subunit has 21 proteins and 1 RNA. That's it. The 16S RNA. Larger subunit, 2 RNAs. Smaller subunit, 1 RNA. This is constant in both. Right, this is constant in both. Now here also if you talk about the smaller subunit of ATS, which is 40S, right, it is made up of 33 proteins. Here 21, here 33. Now the RNA is of 18S. Here it was 16S RNA. That's it. That makes up our smaller subunit of 40S. Now 60S, the larger subunit, would be made up of what? Protein and RNA. है ना? So here what is the situation? Let's see. So here when we talk about 80S ka larger subunit, which is 60S, so here what do we have? We have three types of RNA. 5' RNA, 5S RNA and 28S RNA. And how many proteins? 40 proteins altogether. 40 proteins. There it was only 34 proteins. Yes. So my dear students, when you go for NEET examination preparation, when you go in higher grades, you will have to remember all of this. Now this I have explained you so that you get the justification why 60 plus 40, 100. Right? So 100 is not actually the addition of this thing. It is the weight of this. Right? So 60 plus 40 is not becoming 100 over here. Why not? Because this is not the weight we are talking. We are talking about the sedimentation rate, and that's why it becomes 80. Similarly here, 50 plus 30 should become 80, but no, it is 70. That's just a biology thing. Okay, now let's move off for the function. So what is the function? It is actually actively involved in protein synthesis. That's why we call them as protein factories. है ना? So it has specific sites for the binding of amino acid, for the binding of tRNA, transfer RNA, right, and mRNA as well. Now, it is because of this ribosome, the RER, the rough endoplasmic reticulum, is able to produce protein, to make proteins. Not only that, it also protects the protein chain. Protein chain is what? Protein chain is basically the chain made up of amino acids. The amino acids are linked up together by a peptide bond. This is a peptide bond, and this we will call it as a protein. ठीक है ना? This we will call it as a protein. So this protein chain we have to preserve it also. We have to protect it also. So the larger subunit will bind over this, and that is kind of protecting it for some time. Okay, so now when on an mRNA, what happens? This is an mRNA. Right? mRNA. This mRNA has all the codes required for a particular synthesis of protein. Now, the ribosome binds at a particular set site and then starts synthesizing a polypeptide chain of amino acid called as the protein. Now, is that key one mRNA only one ribosome will bind, so that one mRNA will only produce one little protein? No. Protein can be produced in large amount or less amount, right? So, many ribosomes can be attached, and similar time, multiple protein synthesis can happen, right? And that site where you have mRNA and many ribosomes are attached to it. So this is your mRNA, and to this many ribosomes are attached, and simultaneously they are doing protein synthesis of a particular protein, the target protein which we have got the assignment to produce for. This structure is called as polyribosome. Alright, it's called as a polyribosome. Now, my dear students, challenge as I promised you. Ribosomes are produced and assembled where? Lysosomes, endoplasmic reticulum, Golgi apparatus, or the nucleolus, right? So, let me see where you can answer this or not. You know where to find the correct answer. It's in the Telegram channel. I would post the answer for this thing. Till then, please answer if you can. That's my challenge to you. Keep learning. Thank you for being patient. See you soon.